You know, every single second of every day, there's this invisible war being fought inside your body. It's a chemical battle, really, to keep everything in this perfect, delicate balance against, well, everything you eat, drink, and do. Today, we're going to pull back the curtain on the unsung heroes of this battle, the body's chemical buffer systems.
I mean, really think about it for a second. You can down a glass of acidic orange juice or go for a super hard run that pumps out lactic acid, but your internal environment, it stays remarkably stable. How does it do that? Well, the answer is in this totally fascinating and honestly elegant chemical defense system that we're about to explore.
All right, so here's our game plan. We're going to start with the big picture, the body's balancing act. Then we'll dive into the basic chemistry of what buffers even are. After that, we'll meet the star of the show, the bicarbonate system, and see how it teams up with our organs. We'll also look at what happens when it all goes wrong, and finally, we'll take a little peek into the chemist's toolkit to see how all these ideas are used in the lab.
Okay, first things first, let's talk about the main idea here, which is something called homeostasis. Now, this is the body's incredible, always-on process of keeping its internal environment stable, no matter what's going on outside. It's a constant state of adjustment, and one of its absolute most critical jobs is keeping our body's chemistry right on the money.
This right here, this number is everything. The pH of our blood has to stay within this unbelievably narrow range. If it drops just a little, below 7.35, we're in a state of acidosis. If it creeps up above 7.45, that's alkalosis. And both of these are life-threatening. Why? Because the millions of enzymes that basically run our entire metabolism can only work properly in this tiny, tiny window. Think of an enzyme like a perfectly shaped key for a lock. If you change the pH, you start to bend and warp the key, and pretty soon it just can't do its job anymore. It's a serious tightrope walk, and buffer systems are a safety net.
So, to really get a handle on that safety net, we do need to touch on a little bit of chemistry. But don't you worry, we're going to make this super clear. You've probably seen the pH scale before, right? It's just a simple way to measure how acidic something is. So pure water is neutral, right smack in the middle, at a pH of 7. Anything below 7 is acidic. That just means it has more free-floating hydrogen ions than water. And anything above 7 is basic, or alkaline, which means it has fewer. So our blood, sitting around 7.4, is actually just slightly basic. So what in the world is a buffer? Well, the simplest way to think about it is like a chemical sponge. If you add a strong acid, the buffer just soaks it up, stopping the pH from dropping like a rock. If you add a strong base, same deal. It soaks that up too, and the pH doesn't go flying up. And the way it does this is because it has a chemical tag team ready to go. A weak acid to neutralize any incoming base, and its partner, a conjugate base, to neutralize any incoming acid. It's ready for anything from either direction.
And oh man, this just shows you the power of a buffer perfectly. Okay, look at the unbuffered solution on the left. Starts at a pH of 5, add a little bit of strong acid, and bam, it plummets all the way to 2. Add a little strong base, and whoosh, it shoots up to 12. Just wild swings. Now look at the buffered solution on the right. It also starts at pH 5, but when you add the exact same amount of acid, the pH barely moves, just down to 4.98. Add the same amount of base, and it only goes up to 5.02. The difference is absolutely staggering. That is the stability a buffer gives you. All right, it's time to meet the MVP, the most important buffer system in the entire human body, the bicarbonate buffer system. We do have others, sure, like phosphate and protein buffers, but this is the one doing the heavy lifting. It's the main regulator of our blood's pH, and it's tied into some of our most vital bodily functions.
Now, I know this equation looks a little intimidating, but it's really just telling the story of this dynamic chemical dance that's happening in your blood right now. See, on one side, you've got carbon dioxide, the stuff we breathe out, and water. They get together to form carbonic acid. Then that carbonic acid can let go of a hydrogen ion, that's the acid part, and become a bicarbon ion. And the key is those arrows pointing both ways. It means the reaction is totally reversible and is always shifting back and forth to keep things in balance. It's not sitting still, it's a living, breathing equilibrium. So how does the system know which way to shift? Well, it follows this fundamental rule in chemistry called Le Chatelier's principle. And what it basically says is this: if you mess with a balanced system, the system will push back to undo the mess. So if you add more of something on one side, the reaction will immediately shift to the other side to use it up and get back to that happy, balanced place. It's like chemistry's way of finding its zen.
So this is what our buffer looks like when it's just chillin'. It's in its calm, resting state.
We've got a nice balanced mix of the weak acid part, let's just call it HA, and its partner, the conjugate base, A-. The pH is stable, everything is in equilibrium.
Okay, now let's throw a wrench in the works. We add a strong acid, which floods the system with all these extra hydrogen ions. Immediately, the base part of our buffer, the A-, it springs into action. It just gobbles up those free hydrogen ions, turning them into the weak acid, HA. And here's the magic trick. That dangerous strong acid gets transformed into a harmless weak acid that was already part of the system. The overall pH barely even budges.
And yep, it works just as beautifully in the other direction. If we add a strong base, now it's the weak acid's turn. The HA steps up to the plate. It donates its hydrogen ion to the base, which neutralizes it, and in the process, the HA turns into its partner, A-. So again, a dangerous substance gets neutralized, the balance shifts just a tiny bit, and the pH stays rock solid. It's an absolutely beautiful, self-regulating system.
But here's the really cool part. This whole chemical dance doesn't happen in a vacuum. Our body has this incredible support system to manage it all, turning it into a real team effort between our blood, our lungs, and our kidneys.
First up, the bloodstream is like the highway system, just carrying all the buffer parts wherever they need to go. Then you've got the lungs. They are the rapid response team. Remember how carbon dioxide was on one side of our equation? Well, by controlling how fast we breathe, our body controls our CO2 levels. So if your blood gets a little too acidic, your brain tells you to breathe faster. You blow off more CO2, which pulls that whole reaction to the left and lowers the acidity. It's an almost instant fix. And then, for long-term control, you have the kidneys. They're the master regulators. They're slower, but way more powerful. They carefully decide exactly how much bicarbonate to get rid of in our urine or reabsorb back into the blood, making sure the base part of our buffer is always at the perfect level. So what happens when this amazing coordinated system gets overwhelmed or just fails? Well, as you can probably guess, the consequences are pretty serious. We're going to look at what happens in the body, and then we'll see why these exact same ideas are so critical for scientists working in a lab.
Right, so when the buffer system just can't keep up, you end up with either acidosis or alkalosis. Acidosis is pretty common, actually. In uncontrolled diabetes, for example, the body produces these acidic things called ketones, which can totally overwhelm the buffer. You might have heard of it, diabetic ketoacidosis. It's incredibly dangerous. On the flip side, you can get alkalosis from something like severe vomiting or even from hyperventilating during a panic attack. Both of them are huge medical emergencies because, like we said, our enzymes just stop working right, which leads to total metabolic chaos that can absolutely be fatal if it isn't corrected.
And this need for a stable pH isn't just about our bodies. It's huge in the laboratory. Scientists have to rely on artificial buffer solutions to do their experiments. So if you're trying to grow delicate cells in a dish, you have to mimic the body's stable environment. For that, you might use a buffer called HEPAS, which is fantastic at holding a pH right in that physiological range. But if you're doing experiments with DNA or proteins, you might use a different one, like tris, which works better in a slightly different range. Choosing the right buffer is absolutely critical if you want to get results you can actually trust.
Okay, so for those of you who want to go a little deeper, let's pull the curtain back even further and see how chemists actually design and think about these solutions. We're going to look at two key ideas they use to master buffers, something called capacity and how they calculate it all.
So not all buffers are the same. Buffer capacity is basically a measure of how tough a buffer is, how much acid or base it can soak up before the pH finally starts to move in a big way. Let's go back to our sponge analogy. A buffer that has high concentrations of its acid and base parts has a high capacity. It's a huge absorbent sponge. A really diluted buffer has a low capacity. It's like a tiny little sponge that gets soaked and overwhelmed really fast.
What's so cool about this slide is that it's basically the recipe for a perfect buffer. First, like we just said, you want high concentrations of your tag team. Second, to get the most bang for your buck, you want the amounts of the weak acid and its base partner to be pretty much equal. And third, this one is the secret sauce. You have to pick a weak acid whose pKa is super close to the pH you're trying to hold. You can think of pKa as the acid's natural happy place, the pH where it's perfectly balanced and can fight off changes from both directions with maximum power. When the pH you want matches the acid's pKa, your buffer is at its absolute strongest. And how do chemists put all this knowledge into practice? They use this really elegant tool, the Henderson-Hasselbalch equation. This formula just beautifully connects those three critical things we were just talking about: the pH you want, the pKa of your acid, and the ratio of the base to the acid. It lets scientists calculate the exact amounts of everything they need to mix together to create a buffer with a super precise pH. It is easily one of the most practical and important equations in all of chemistry and biology.
So in the end, the ideas we've talked about today aren't just happening inside of us. The world's oceans, for example, have their own gigantic bicarbonate buffer system that absorbs carbon dioxide from the atmosphere and keeps the pH stable for all marine life. Buffer systems are just this fundamental chemical principle for creating stability. It kind of makes you wonder, right? What other invisible chemical systems are quietly humming along in the background, maintaining the delicate balance that allows our world and our own bodies to exist?